Hello, welcome to the Science Guide ASAP Learning. Are you struggling to get an A in biochemistry? Don't worry, you're not alone. In this video, I'll share some tips and strategies that will help you get an A in biochemistry. Master the basics. Biochemistry is a subject that builds on the foundational knowledge of chemistry and biology. Therefore, it's essential to have a solid understanding of basic concepts such as chemical bonding, acid-base reactions, and cellular biology. Reviewing these basic concepts before diving into biochemistry can help you better understand the more complex material. Attend lectures and take notes. Attending lectures is crucial for success in biochemistry. Instructors often provide valuable insights and explain difficult concepts in ways that are easier to understand. Taking detailed notes during lectures can help you retain the information better and serve as a useful reference when studying for exams. Stay organized. Biochemistry is a complex subject that involves a lot of information. Staying organized is essential for success in this subject. Create a study schedule, keep track of important deadlines, and organize your notes and study materials. Practice, practice, practice. Biochemistry is a subject that requires a lot of practice to master. Practice problems, worksheets, and quizzes can help you solidify your understanding of the material. Work through as many practice problems as you can and seek help from your instructor or peers when you encounter difficult concepts. Form a study group. Studying with peers can be a helpful strategy for success in biochemistry. Form a study group with classmates who are also interested in getting an A in biochemistry. This can provide an opportunity to discuss difficult concepts, ask questions, and learn from one another. Seek help when needed. If you're struggling to understand a concept or are falling behind in the course, don't hesitate to seek help. Instructors and teaching assistants are available to provide guidance and support. Additionally, tutoring services and academic support centers can offer additional resources and assistance. In summary, getting an A in biochemistry requires dedication, hard work, and a solid understanding of the material. By mastering the basics, attending lectures, staying organized, practicing, forming a study group, and seeking help when needed, you can achieve success in this challenging subject. Bonus. Here are some high-yield questions that professors love to ask. A protein contains 20 amino acids. How many different possible combinations of amino acids can be used to make a protein that is 100 amino acids long? Asterisk. Pause this video and solve asterisk. Answer. There are 20 possible amino acids that can be used at each position, so the number of possible combinations is 20 to the power of 100, or approximately 1.27 times 10 to the 130. Explanation. This question tests knowledge of the combinatorics of amino acid sequences. There are 20 different amino acids that can be used at each position, so the total number of possible combinations is the product of 20 for each position in the sequence. A researcher wants to determine the concentration of a protein in a sample. The protein has an absorbance maximum at 280 nanometers with an extinction coefficient of 1.2 times 10 to the 4 m⁻⁻1 cm⁻⁻1. The sample has an absorbance of 0.5 at 280 nanometers in a 1 cm cuvette. What is the concentration of the protein in the sample? Asterisk. Pause this video and solve asterisk. Answer. The concentration of the protein in the sample is 2.08 times 10 to the -5 m. Explanation. This question tests knowledge of Beer's law and protein quantification. According to Beer's law, absorbance is proportional to concentration and path length. The concentration of the protein can be calculated using the equation A equals epsilon LC, where A is the absorbance, epsilon is the extinction coefficient, L is the path length, 1 centimeter in this case, and C is the concentration. Rearranging the equation to solve for C gives C equals A, epsilon L. Plugging in the values gives C equals 0.5, 1.2 times 10 to the 4 m⁻⁻1 cm⁻⁻1 by 1 centimeter, equals 2.08 times 10 to the -5 m. In glycolysis, the enzyme aldolase catalyzes the conversion of fructose 1,6-bisphosphate into 2,3-carbon molecules. What are the names of the 2,3 carbon molecules produced? Asterisk, pause this video and solve asterisk. Answer. The 2,3 carbon molecules produced are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Explanation. This question tests knowledge of the enzymes and intermediates involved in glycolysis. Aldolase catalyzes the cleavage of fructose 1,6-bisphosphate into 2,3 carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These two molecules can be interconverted by the enzyme triose-phosphate isomerase to yield 2 molecules of glyceraldehyde 3-phosphate, which then enter the next steps of glycolysis. Hope this helps. Please support this new channel by liking this video and subscribing for more videos like this one.